Thank you. I'm delighted to be back here. The last time I spoke at this podium was 18 years ago when I was taking an undergraduate seminar in immunology, and at the end of the course we had to give our presentations. It's fun to be back. And I'm also pleased to be talking to science teachers in high school. I still fondly remember my science teachers in high school. Phil McRae, who taught biology at New Trier High School in the Chicago suburbs, and Doc Wagner, who did chemistry, and Alan Bricks, who taught physics. They were wonderful instructors, and I failed them, of course, by not proceeding to a laboratory-based career. I do clinical medicine principally, but I have great admiration for all you do, and I'm actually pleased to be here to share a few thoughts with you today. My talk today is really going to be about the clinical interface between modern science and clinical medicine in my own discipline, which is breast cancer. And the simple message I want to leave you with is as follows. For the first time, we are taking care of disease based on the biology of that tumor type. And so it is imperative that doctors and more patients understand something about biology to understand the type of the treatment they're going to get. In cancer medicine, for the past several millennia, the principal form of therapy has been to surgically cut it out. And you don't have to actually understand very much biology to know that those were your options. And it's only about the past 100 years that that was even something you would ever consider doing. But now we are truly entering an era where biology is driving clinical medicine. It's not just about understanding at a very sophisticated level how this came to be and providing explanatory power, but it's really about making concrete real-world decisions with individual patients that's going to help them live better and longer. Dr. Brugge, my very distinguished colleague, is going to talk to you in the second hour about how women get to this point of having breast cancer and other basic transformation steps. But again, my talk is going to be more clinical and really focus on two paradigms where there are some nice stories about how we're using molecular medicine to inform clinical decision making. Now, the first thing to point out is this is a clinical talk, therefore the PowerPoint background slide is blue. As you may have noticed, all laboratory and basic science talks have background slides that are white. All clinical slides have backgrounds that are blue. I do not know why that is the case, but it is absolutely a true law. See if that holds up in your training. Well, you know, we actually learn a lot about breast cancer when a woman goes for surgery. We find out how big the tumor is, whether or not the lymph nodes are involved with cancer. We measure the hormone receptor status, that is to say the estrogen receptor and the progesterone receptor in the tumor. And the pathologist can also tell us the tumor grade. And we also are increasingly measuring the expression of a protein called HER2, which stands for the human epidermal growth factor receptor 2. And then there are a variety of other pathological features which over the past 20 years people have looked at and principally discarded as being important measures of clinical activity. So we learn a lot about the clinical presentation, how big it is, how far it has spread, and a little bit about the biology of the disease for all women already in breast cancer. Now, breast cancer already is years ahead of most solid tumors. There are no other solid tumors that we treat, colon, pancreatic, prostate, ovarian, where they even bother to measure these biological growth factor receptors because, in fact, we have no therapies that are targeted against them for other tumor types. So breast cancer is the queen of the medical subspecialties in oncology because we can take advantage of them. And the paradigm that has been most important in treating breast cancer has been manipulation of the endocrine environment using estrogens or anti-estrogen therapies in women whose tumors overexpress estrogen receptor, which is the vast majority of breast cancer, probably 2/3 of breast cancer. But that's not what I'm going to talk about today, even though it does represent a classic example of molecularly guided therapy. I want to take you the next step now into looking at the molecular heterogeneity of breast cancer. Now, as clinicians, I know that breast cancer is a very heterogeneous disease. I will see 300 patients or so this year with breast cancer, and while things like the tumor size and the nodal involvement do predict how well a woman is going to do, there's actually tremendous heterogeneity. So most node-positive breast cancers probably won't recur, but some will. The vast majority of node-negative won't recur, but some will. And we're actually pretty bad at figuring out who is going to be which way things are going to go. So to advance this field, many laboratories, including those at Harvard, and this particular data set from David Botstein's laboratory at Stanford, have undergone molecular phenotyping of breast cancer. Joan and others, I'm sure, have explained more to you about how this is done, but essentially you take RNA from tumor specimens and you create an RNA expression array using more than 8,000 genes on these chip arrays. Have you guys heard something about chip arrays? Yeah, okay. And I know nothing about chip arrays besides the next two slides, so please correct me when I misspeak. They then use computer analyses to identify 427 genes that differ between the tumors, but not from paired specimens obtained in the same tumor. And finally, they settled on about 80 genes that could be used to cluster these tumors in an algorithm built around prognostic information. And this is what they end up with. Now, what you see in this depends on what you look for. Mostly my colleagues see grant money, because anybody who's doing gene chip arrays is instantly fundable to an inordinate degree. Otherwise, it's kind of a Rorschach blot, and you need big-time computing and statistics to really understand these. But suffice it to say that with clustering techniques, within the spectrum of breast cancer, they could identify somewhere between 5 and 6 major categories of tumor type. Now, that's an important number for us as clinicians, because on the one hand, every single column here, which each one is a different tumor from their set, is a little bit different when you look at all these hundreds of genes. So on the one hand, everybody's tumor is a little bit different from the next person's tumor. But if they're all totally different, you can't treat them, because we can't develop therapies for individual we just don't have that kind of resources or technology. But if there are a handful of different kinds of breast cancer, well, then that's something useful, because when there are 200,000 cases of breast cancer every year in the US, and if you divide them up into 4 or 5 major subtypes, that's enough material that you can begin to build diagnostic and developmental drug programs around those individuals. The other nice thing about this clustering was that it confirmed information that our humble clinicians knew all along, namely that estrogen receptor positive versus negative is the first big cut. That's the first separation, and that remains the single most important distinction. That was confirmed by this very fancy biology. But the other interesting point was that particularly within this estrogen receptor negative family of breast cancers, there were a couple of other distinct types. And one of those types was the so-called HER2-positive breast cancers, which line up here. They tend to have overexpression of a variety of epidermal growth factor-like proteins and relative underexpression of some other markers. And finally, there is the so-called basal-like or basaloid types of breast cancers, which also emerge as a distinct molecular phenotype. These particular tumors tend to be estrogen receptor negative and to lack the overexpression of the HER2 oncogene. And it also turns out, which is relevant for the second half of my story today, that these basal-like or basaloid tumors are particularly enriched in women who have hereditary predisposition to breast cancer, with the BRCA1 mutation in particular, which commonly gives rise to this basaloid type of breast cancer. So on the one hand, this data has proven enormously powerful in allowing us to begin to segregate breast cancers based on biology rather than just on looking under a microscope or doing some relatively crude immunohistochemical stains. On the other hand, it has also reified some important clinical observations, namely that estrogen receptor defines different biological subsets, and that this HER2-positive subset of breast cancer is a unique beast. Now, this information is prognostically important. This is the same data set. Women whose tumors are HER2 positive, shown here in purple, or those basal-like ones, shown here in red, tend to be at greater risk for recurrence over time. Women whose tumors are estrogen receptor tend to do very well in comparison. And interestingly, there was a second group of ER-positive breast cancers, here shown in the light blue, which actually did very poorly, even though they were ER-positive. And this is the kind of information that we are looking for, because we want to be able to stratify people into very good or not so good groups, because we can direct therapies at them. Well, let me tell you a little bit more about the HER2 story. It's one of the great stories in translational medicine right now. The new oncogene was originally defined in neuroblastoma cell lines. That's why it's called new. That were caused in rats by exposure to ethyl nitrosurea. So this is not a human tumor. This is a chemically induced murine or rat carcinoma. And in 1981, some famous experiments done at MIT showed that DNA from neuroblastoma cell lines could in fact transform a mouse fibroblast, the NIH3T3 cell, from growing in a tissue monolayer into growing into heaped up piles of cells that resembled a tumor. And in 1984, they were able to winnow the DNA down to show that this new oncogene was the transforming property, and that this was homologous to a related protein called ERB2, the human epidermal growth factor receptor 2. And in 1987, Dennis Slayman and others working at laboratories at UCLA showed that HER2/neu gene amplification was associated with a poor prognosis in breast cancer. So they had this molecular probe, and they looked at about 200 cases of breast cancer and showed that those women whose tumors had genetic amplification as an acquired gene defect, not as a baseline or hereditary gene defect, but as an acquired gene defect, did less well in the long term than did women whose tumors lacked this. And what has followed has been a huge amount of research, which has now shown that overexpression of the 185 kilodalton HER2 protein arises from gene amplification of this gene complex on human chromosome 17, that this tends to be associated with other adverse prognostic factors in breast cancer, that is, lack of hormone receptor expression, high-grade, rapidly growing tumors, early sites of recurrence. This is relatively rare. It happens in probably something like 20% of all cases of early-stage breast cancer. But because it's an adverse prognostic factor, it is somewhat more likely to be seen in women with metastatic disease who have recurrence, and therefore maybe 25 or 30% of such women would have HER2 gene amplification in their tumors. A variety of studies, this group happens to be just one report from Australia, have shown that HER2 gene amplification, no matter how you define it, is associated with poorer outcome in breast cancer. So there's a big controversy as to how best to test for HER2, whether you use staining methods or whether you use gene methods looking for copy overexpression. But suffice it to say that no matter how you define it, HER2 negative tumors, shown here, do better than HER2 positive breast cancers, which always have a greater risk of recurrence. Are you guys familiar with Kaplan-Meier survival type curves as I'm showing here? Have you seen this before? This is essentially what they call a life table curve, where you start at 100% free of cancer, and the curve plots out the risk of recurrence over any given time period. So the steeper the curve, the greater the risk of recurrence through any period of time. And HER2 positive tumors do less well. And the other thing we've learned, and Joan is going to fill in all the details about what happens here for you, but in brief, as breast tissue transforms from normal tissue through abnormal growth to what they call hyperplasia, where the nuclei begin to look abnormal, to a precancerous lesion in the breast called ductal carcinoma in situ, and finally to invasive breast cancer, there are a whole host of accumulating genetic and epigenetic changes and changes in response to growth factors, and others will explicate this. But the point for today's discussion is that gene amplification for the HER2/neu oncogene probably happens somewhere right around this DCIS stage. And if this does not happen, you can still get breast cancer. In fact, 80% of breast cancers lack this, but you will never go back and reacquire amplification of the HER2/neu oncogene. By contrast, if this event arises in the DCIS stage, this gene amplification of the HER2/neu oncogene is fixed and becomes a part of that breast cancer for the life of that tumor.
Let me ask a question. The AGR2, the normal tissue, is that sort of asleep and not working?
It's actually expressed on a lot of normal tissues, probably at the level of around 20,000 copies per normal breast tumor cell or breast tissue cell. When you have the gene amplification, you get 2 million copies. So a 2 log order increase in the level of surface expression of the receptor because of the gene amplification that has arisen for that oncogene. So yes, it is a normal component of the body. We don't know exactly what it does, but it probably is involved in a variety of growth and, signals in both the breast and elsewhere. Everybody has it. Every tumor has it. A subset of tumors have gene amplification, which gives rise to huge amounts of expression of that protein on the cancer cell. Other questions? So this just is to remind us that this is a seminal event if it happens, and then the primary tumor would be HER2 positive throughout the life of the patient. By contrast, if it doesn't happen, once you actually reach the primary invasive tumor stage, you're going to be biologically distinct for the rest of the tumor's life. And again, another schema just to suggest that this happens somewhere as tissue transitions from a hyperplastic, benign growth towards an invasive cancer, probably at the level of this in situ cancer in the breast. Well, it was asked what these things are doing. So actually, this is a member of a very large family of growth factor receptors, all of which share tyrosine kinase domains intracellularly and homologous features extracellularly. And other members of this family include the epidermal growth factor receptor, which binds to EGF. It also binds to a variety of paracrine and endocrine factors. This has been an area of intense drug development because almost all human tumors that you can think of, like lung cancer and prostate cancer and head and neck cancer and things, express some degree of this EGF receptor, which has made it very appealing for the drug companies to go after. HER2, interestingly, has not had an identified ligand as yet. And it is thought that the biological role of this protein is to bind either to the EGF receptor or to these other family members in heterodimerized protein complexes to generate an intracellular signal. And then there are these other members of the family, HER3 and HER4, each of which bind these related products called heregulin or neuregulin. So a related family of growth factor receptors which generate tyrosine kinase signals and which normally probably bind to one another to moderate these endocrine and paracrine type effects in the body. This is a gross schema of what might happen when you have binding of an EGF family ligand to the ErbB receptor. You then generate signals which interact with a host of different pathways. I used to know more about these pathways, but I confess I've actually forgotten all of it. But others can tell you how important they are at regulating normal cell growth physiology. This is my appreciation of this pathway in general. And the point is there's a lot of overlap and redundancy in these various pathways. Now, all of this would have been well and good, except that it lent itself to the development of a targeted therapy. And that drug we now call Herceptin, Herceptin, which has the generic name trastuzumab. As you may know, FDA has asked that all these humanized antibodies have the UMAB for human antibody ending. So that's why there's a lot of drugs that are coming along with UMAB endings. And what investigators at UCLA and then clinical scientist development people at Genentech did was to make a mouse antibody against the HER2 protein. So they took buckets of human HER2 protein and injected the mice and made mouse antibodies against human HER2, and then they cloned these proteins, these antibodies. And then they went in the lab and they took all the mouse structural elements of the immunoglobulin and they replaced it with human DNA, which is called humanizing it, so it doesn't cause antibody responses to the foreign mouse protein. So all that's left of the mouse protein in this drug are the yellow specificity determining regions of that antibody. Way out at the tips of the antibody is mouse, and everything else is human through recombinant DNA technology. And then they transfect this stuff into Chinese hamster ovary cells, which are really just factories of protein production, and they grow them up in huge copper vats like beer fermenting. And it actually smells like beer fermenting because it's all in this very rich medium, and it really is sort of a yeasty kind of smell, though of course it's all very sterile. And these things just sit there, and these transfected cells just crank out buckets and buckets and buckets of protein, which is then purified through whatever strategies they use to purify the protein. And you're left with essentially a pure monoclonal antibody that began its life as a mouse product and still has a little bit of mouse protein on the outside, but which binds to the human HER2 protein and which doesn't cause immune reactions in people because everything else has been replaced by human DNA. Does that make sense? Yeah.
Why did they leave the mouse stuff? Isn't that the most critical part?
Well, that's what binds to the human protein. So you need the mouse stuff out there because that's what tells this antibody to glom on to the human HER2 protein. You actually can't get it because we all have innate tolerance of our own growth factor proteins. And so you can't easily get humans to make an antibody response against our own self proteins, if you will. Essentially what you do is you end up, you make this monoclonal antibody from the mouse, and then you have, they know on the antibody gene where the parts are which determine where it binds, and where the parts are which are just these structural sort of framework pieces. And then they clone out the mouse structural pieces and replace them with human DNA. It's fantastic technology. And this has now been done for a number of drugs in oncology. There's another one called Rituxan, which binds to the protein called CD20, which is on many lymphoma cells. So it's another way of humanizing these antibodies. And this has become a actually surprisingly successful commercial effort. Yes, it's, I mean, I don't know which it is, but it's a clear IgG1 heavy chain and kappa chain structure. So they could tell you exactly what it is to the base pair if they wanted to. Yeah?
HER2 and NEU, are they identical or is one a mutated form?
No, in common parlance, they're essentially identical at this point. The name has evolved as it's gone from something that scientists understand, where you have italicized gene names and fancy things, to something primitive so clinicians like me can understand it. So we just call it HER2. And of course, it was sort of cute because it was a women's cancer, things that are her too, you know. So, okay, and so they've actually now gone, and you can do very careful structural analyses to actually show where Herceptin, which is what this antibody is now called, this is Herceptin now as a trade name. And you can show where that actually binds sort of down near the, this would be the extracellular domain right here coming up, and it binds down towards the cell membrane of the human HER2 protein. And if you want, you can even take this to incredibly refined detail, where in crystal structures, you know, here's the surface of the antibody coming up and binding right up against to the HER2 protein that's shed into the serum. So it's really an incredibly specific, targeted therapy which only recognizes that HER2 protein, which is expressed on many, many normal tissues, but only overexpressed in the subset of breast cancer. Well, what happens? So here's a breast cancer cell, and you can identify this as a breast cancer cell because it's pink. And work with me here a little bit. So this is supposed to be the genetic amplification of the HER2 new oncogene. There are multiple copies. You know, normally there would be two, one from mom and one from dad, and here at least there's a doubling of that number. And that leads to overexpression of this 185 kilodalton HER2 protein on the surface of the cell. And this antibody can be given to people. It floats around. It binds to this HER2 protein on the cell, probably initiates a cross-linking event, which somehow generates intracellular signals, which I imagine literally and figuratively as a black box inside the cell. But suffice it to say that this signal actually changes the growth patterns of the cancer cell. It slows down the rate of growth, it induces apoptosis, programmed cell death, and it potentiates the effects of chemotherapy. Now, then this went through an enormous amount of clinical development. And I confess, coming from a science background, I didn't really understand how drugs were developed when I went into clinical oncology. And it turns out that the easy part actually is getting the drug, and the hard part is showing that it's safe and effective and useful in people. And I say this not to be some kind of apologist for the drug companies and the outrageous prices they charge everyone and things like that, but I mean, it turns out that if you want to work in oncology, you got to work with the drug companies because they have the drugs. It sounds silly, but that's actually true. Academic centers and universities have been absolutely dismal at developing drugs. They don't have the time frame and they don't have the resources. It probably costs $1 billion to develop a drug in the current market. And so it's not something that academic centers do very well, so we have to work a lot with the drug companies. As the old Willie Sutton joke was, you know, why do you rob banks? That's where the money is. It's sort of true, why do you work with the drug companies? That they have the drugs. Nobody in this building has a single drug, nor will they in the next 10 years. It's just not something they're interested in developing. So to develop it, they first gave this drug to people to see if it was safe, and in the limited experience, it seemed to be safe. Just to remind you of, oh, sorry, I'm going the wrong way here. So we go backwards, I guess. There we go. this drug is this antibody. So when you infuse it into people, you put an IV into their arm, and then you drip it into their arm as a reconstituted solution. It takes about an hour to give it to a person. And about a third of the people on their very first dose will have a low-grade reaction, like with chills or a fever, because you're infusing a protein into them, which they don't tolerate perfectly. But most people do very well. And actually, when you give it by itself, then there are hardly any acute side effects. So this is not chemotherapy. It does not make your hair fall out. It does not make you sick to your stomach. It does not lower your blood counts. It does not make you tired. It's a monoclonal antibody. The other implication, which we're only still wrestling with, is this actually, in comparison to most drugs, monoclonal antibodies are very stable. I mean, you know, the body has antibodies and they last for decades, right? You know, you've got a tetanus vaccine and, you know, you've got tetanus antibodies for the rest of your life. So these things are incredibly stable in the body. They last at levels for months and months and months, which is unusual for drugs. Okay, so the study that really established this drug's usefulness is a randomized clinical trial that was published 2 years ago in the New England Journal of Medicine, where women who had this particular kind of breast cancer, HER2-positive breast cancer, which meant we had to test everybody for this, because usually we just treat breast cancer the same way, but now we had to take the tumor, test it to see if it was HER2-positive, and then they were given chemotherapy, and I won't bore you with the details of it, but they were given either chemotherapy alone, which was either two flavors, doxorubicin and Cytoxan AC, or Taxol, paclitaxel, plus or minus the addition of trastuzumab, that's Herceptin. So again, a randomized trial where everybody gets chemotherapy and half the patients receive Herceptin in addition to chemotherapy. And the major finding from that trial was that adding Herceptin to chemotherapy dramatically improved the time to treatment failure, or the so-called progression-free survival, for women with metastatic breast cancer. So with chemotherapy alone, most of the women, half of the women, had progressed within 6 months of starting chemotherapy treatment. That is, their tumor was overtly growing within 6 months of beginning chemotherapy. And by around 12 months, all but about 10% of the women's tumors were progressing. By adding Herceptin to chemotherapy, we shifted out this curve such that the median time to progression was more like about 10 months, and at 1 year, something like a third of the women were still free of cancer recurrence. Now, unless you go to a lot of oncology meetings, you're looking at this and you're saying, that doesn't seem like a lot. And on the one hand, you're right. On the other hand, in terms of clinical advances in cancer, this is as close to a home run as you'll ever see. I mean, this was a dramatic change in the natural history of a very aggressive form of breast cancer. And it also increased the chance of the tumor shrinking, you know, responding to the treatment. So in the group that got chemotherapy alone, about 25% of the women had overt shrinkage of the tumor. By adding Herceptin, you took that up to something like 50 or 60%. So you doubled the chance of the tumor shrinking, and you prolonged their disease-free survival. And eventually, that translated into an improved overall survival. Again, here is whether or not the patient is alive or deceased. Moving forward, within 2 years, half the patients given chemotherapy alone have passed away, and you extend survival by an average of about 6 months by introducing this drug. Now, 6 months is not curing their cancer, but in fact, this represents, as I said, a fairly dramatic difference in oncology products. And this is the kind of thing where you could just sit in the back of an oncology symposium and look at the data, and you knew that the drug would be approved by the FDA and would be actually something people would use all the time. Now, the other surprise in this is that Herceptin increased the risk of heart damage when women were getting chemotherapy at the same time. And that is still a relatively unexplained phenomenon. However, it seems that the protein's also on the heart. So this HER2 protein is also expressed in heart tissue, and it may be that Herceptin exposure interferes with the normal repair that the heart is always undergoing. So heart cells come and heart cells go, and they're always turning over. Herceptin blocks some of that recovery, and it seems to potentiate the heart-damaging effects of certain kinds of chemotherapy. That was a totally unexpected surprise, and people weren't really ready for it, and we've had to, use different chemotherapy regimens to avoid that. Well, where has this led in terms of scientific advance? As recently as 1980, there were less than 10 papers written on this protein in the clinical literature. As of 2003, they're being published at a rate of about 1,000 per year. So this has been an extraordinary advance. And it has changed the way we take care of metastatic breast cancer. It has meant that we have to test all women who have metastatic disease to see whether or not their tumor is HER2 positive, because if it's HER2 positive, we're going to offer them this drug. And I won't show you the slides, but it turns out if you're HER2 negative, there's no clinical advantage for taking this product.
What percentage are HIV?
About 20% of all breast cancer and about 25% of metastatic breast cancer. So it's a 1/4 to 1/5 of all women with breast cancer. There have been new toxicities introduced, especially heart failure, which has meant we've had to work around that. This has been, it's meant a lot of other companies are interested in other drugs that will target the HER2 protein. So maybe a small molecule that you could take by mouth might do the same thing. That's something that's intensely being developed. There are now big international and national trials going on to look at this drug for early stage breast cancer. That is, not when women have recurred with their breast cancer, but when they've just been diagnosed. Can we introduce this drug and help them prevent that ultimate breast cancer recurrence, which can be a life-threatening problem? It's also meant that we are probably, within the next couple of years, going to segregate HER2-positive breast cancer away from all other types of breast cancer in terms of our treatment paradigms. So women who have HER2-positive breast cancer will sort of go down one path, and HER2-negative breast cancer will go down these other paths. And that has a lot of implications for how we evaluate and treat people. So I realize that the data are limited, but you cannot underestimate how, in the cancer field, this has radically revolutionized our approach to the majority of women now being diagnosed with breast cancer, either because they have metastatic disease and we're going to treat them, or because they have early stage disease and we're actually looking to see if this drug is going to help further prevent their recurrence. Again, the theme being, you have to know something about the biology to appropriately select patients. Now, you can imagine that this takes a fair amount of explanation to patients. You know, it's not that you say, Mrs. Jones, you have breast cancer, I'm going to give you this. You say, well, Mrs. Jones, you have this kind of breast cancer. And those treatments over there don't work for you, but this treatment over here does work for you. And that presumes a certain amount of scientific literacy. And you know, we actually, this is a new problem for us because most of our patients don't have that much scientific literacy. Most of our doctors don't have that much scientific literacy, but many of our patients don't as well. So let me transition then, with no particular segue, to hereditary breast cancer, because I think it's another instance where we are now using molecular science to help guide treatment for patients. As you may know, there are genes that increase the risk of getting diagnosed with breast cancer. The most famous of these are the so-called breast cancer 1 and 2 genes, which are actually on different chromosomes. And women who harbor a genetic defect in one of the BRCA1 or 2 genes, these are autosomal dominant genes, are at substantially greater risk of getting breast cancer than women who don't. Just to put that in some perspective, there's a lot written in the popular press about hormone replacement therapy increasing the risk of breast cancer, obesity, reproductive habits, when you had your children, alcohol consumption. Those are all established risks for breast cancer. However, the relative risk for most of these is incredibly low. That is to say, you know, women who had a hormone replacement therapy had something like about a 20 or 30% chance greater than the average woman who never took hormone replacement therapy of getting breast cancer. By contrast, women whose tumors are BRCA1 and BRCA, or women, excuse me, who have BRCA1 or BRCA2 mutations in their germline, these are hereditary germline transmitted DNA damages, are at 10 times the risk of getting breast cancer compared to women who do not have them. Said another way, the lifetime risk for the average American woman is that 12% figure, you know, 1 in 8, which is frequently discussed in the press, of developing breast cancer. A woman who has a BRCA1 or 2 mutation would have probably an 80% lifetime risk if she lived to be 80 years old of getting breast cancer. It is a 5 to 10-fold increased risk. Yeah.
This may be a little bit out of left field, but there's been some talk that's been controversial about an increased risk of early, I think between 9 and 13-week abortion and breast cancer. Is there any documentation or is there like a ballpark mechanism?
That literature is highly contaminated by the political process. There has been some very limited retrospective observational data that women who had abortion might be at slightly greater risk. To the extent they were at greater risk, it was on this order of magnitude. It was one of these very tiny treatment effects. These reports have been very inconsistent. They have not been supported by all studies. In fact, the largest studies have not suggested that this is a strong risk. It is confounded because of the fact that many women who get abortions are also more likely to either take hormone replacement therapy or get birth control pills for understandable reasons. But mostly it has been circulated endlessly in the popular press by a political group that is using this as a reason for arguing against getting abortions.
Family history versus the mutation factor.
So everybody has a family history of breast cancer. I won't make women in the room raise their hand, but if you do this, it turns out that probably 2/3 of you know someone in your greater family who's had breast cancer. It's a fairly common disease. And the point here is to distinguish a family history, which is my dad's Aunt Polly had a cousin who I think had breast cancer when she was 79 and in the nursing home, from a real inherited predisposition where there's strong, clear evidence that there's a familial defect that's giving rise to this. And we'll come back to that in a second. Now, fortunately, the vast majority of breast cancer is not heredity. So in breast cancer, probably 5 to 10% of all breast cancer is truly genetic. That is, there's an obvious gene defect that gives rise to breast cancer. Another 15% or so, and this may be an upper estimate, tracks into families where there seems like there's a lot of breast cancer. We can't identify a specific gene, but it kind of smells to the geneticist like there's an element, something going on here. However, 75 to 80% of breast cancer is not hereditary breast cancer, even though most of these women, again, will have a great aunt or a second cousin who did have breast cancer. That's not what we're talking about. For ovarian cancer, it's even smaller as a slice of the pie. Most ovarian cancer is sporadic as opposed to hereditary. However, for those women who have these mutations, the risks are very high. Now, what are some of the features that would suggest you have a true hereditary breast cancer? Well, obviously, if someone in the family has been tested and you know that they have a defect in BRCA1 or 2, which is increasingly the issue, then they are at risk. Families which have both breast and ovarian cancer, because it turns out that these genes give rise to both breast and ovarian cancer. One of the many fundamental mysteries we don't know about these genes is why they don't get pancreatic cancer and lung cancer and prostate cancer and esophageal cancer and all the other afflictions of cancer medicine. Why in particular you get breast and ovarian cancer. We don't have good data on that. It certainly may have something to do with the fact that these are tissues that undergo secondary development during puberty and the reproductive life of a woman. So whereas most tissues that line the body in the lung and in the gut sort of do their thing from the moment you're born till the time you die, actually the breast and the ovarian tissues undergo secondary development with puberty and then further changes if the woman becomes pregnant and with each menstrual cycle. And so that sort of ongoing stimulation may have something to do with the risk. Families where there's male breast cancer, which is very rare. Families in which 2 or more family members had breast cancer as younger women, less than age 50. Or families where one family member had breast cancer less than age 50 and there is Ashkenazi ancestry. There are certain founder mutations, so-called, which seem to be tracking in the Ashkenazi Jewish community. Or ovarian cancer plus Ashkenazi ancestry. These Ashkenazi ancestry is principally what the population scientists would call, an island effect or a founder effect. That is, a relatively large group of people that just inbred such that certain mutations were carried along with them. There's no other particular issue. I once joked at a tumor board, if the person converts, do they still have the same risk of breast cancer? They obviously do. Oh, come on, that was pretty good. All right. So the prevalence of these genes in the general population is very low, about 1 in 1,000. By contrast, it's about 2% in the Ashkenazi Jewish population. Amongst women who have breast cancer, about 3 to 5% of all women have a BRCA1 or 2 mutation. Only about 10% in the Jewish community. We actually, because Judy Garber, who is the lead geneticist in the cancer program at Dana-Farber, has a very large population of women in Boston. We see a lot of these people, but even there, it's only 10% risk. Young women with breast cancer, still only a 10% risk of having a true genetic predisposition. And women with ovarian cancer, again, walking in the door, about a 10% risk. So on the one hand, we clearly can identify factors that increase the likelihood of this breast cancer being a genetic tumor. On the other hand, it's still the exception, not the rule. These are just lifetime estimates for developing cancer in women who have these gene defects. And again, the point is it's very high. If you have, in various studies, hereditary BRCA1 or 2 mutation, you have somewhere between a 60 and 80% lifetime risk of developing breast cancer if you live to be 80 years old, and something approaching a 40% risk of developing ovarian cancer compared to this 12% number for the general population. And these are some of the most recent data from Mary Claire King, a geneticist at the University of Washington, who has really been a pioneer in this field, looking at the lifetime risk. And she makes the argument that when you really identify cohorts very carefully, if you live long enough with these genes, essentially you have nearly 100% risk of getting breast cancer. Now that might not be until you're 100 years old, but starting anywhere between age 30 and 40, there is a very rapid and steady increase in the rate of developing breast cancer. She has some very provocative data also on this slide that women who are physically inactive are at somewhat greater risk than women who are physically active. Whether or not that holds up in larger studies remains to be seen. But she makes the point that this is an interesting observation that suggests an interplay between environmental factors like exercise and hormonal milieu and genetic factors which give rise to an increased risk of breast cancer. This is more of the same. So what are these defects? Well, there are 2 genes. They are both very large and complicated genes. And in the Ashkenazi Jewish population and in most of the other population, there are a few specific mutations that give rise to these predispositions. One is the 185 del AG, which essentially is a single deletion of a single base pair in this protein. Another is an insertion of a C way out here, which gives rise to a deranged protein. And in the BRCA2 locus, again, a single point mutation. So these actually can be tested in an Ashkenazi family very easily because all they do is test a panel that looks for those 3 mutations. For non-Ashkenazi families who want testing, they have to sequence the whole gene, which is actually an enormously complicated, very long gene and is much more technically demanding. This audience is familiar with this, but these are all arising from point mutations. So if you have a normal sentence of DNA, you can get a missense mutation, as you know, which changes a word. But what we're mostly seeing are nonsense or frame shift mutations, which end up either terminating the protein or giving rise to an aberrant protein, either through deletion for a frame shift or through insertion. The point is, single point mutations are giving rise to abnormal proteins. So what's happening? I think this was given for a class which was like some men's health thing, so they expressed this as whether a car would function. I don't know what the metaphor was, but I stole these slides from one of my colleagues. Normally, you have 2 functioning BRCA1 proteins in the typical cell, and the car is firing on all cylinders, if you will. If you inherit a defect, you end up with a non-functioning protein on one of those alleles in chromosome 17, and you're left with only one functioning protein. That's probably enough unless the tumor cell undergoes loss of heterozygosity. So if you lose somehow your other normal BRCA, which happens at some finite rate, then you're up a creek because you have one non-functional protein and you've lost your functional protein. And at that point, you slam into the wall with risk of cancer going way up. I'm not going to elaborate what these proteins are doing, except to say that they both, and Joan may address this in greater detail, are part of a very large complex which seems to be involved at some fundamental level with DNA repair. And if you lose function of either BRCA1 or BRCA2, you end up with bad DNA repair, which is necessary. DNA mutations accumulate within cells, and that predisposes you to malignancy. Now, it turns out that in all of breast cancer genetics, the BRCA1 and 2 genes are probably about half the story. That is, you can find families where you're looking at this family pedigree, mom had cancer, her mom had cancer, the sister, you say it's got to be something obviously genetic. And when you actually do the testing, you don't find a BRCA1 or 2 mutation. That happens about half the time. And there are some rare other familial tumor syndromes, but much of it is probably just undiscovered genes, which have either a low penetrance or are less easy to identify than the BRCA1 and 2 genes have been. Okay, so again, I promised you I would take this into clinic. The matter arises, we can test for this. And how do we think about this for women who have breast cancer or women who do not have breast cancer? Well, there are a bunch of questions. The first is, fundamentally, do these tumors behave differently than non-BRCA breast cancer? Should our treatments be different? How can we prevent these tumors? And who should be tested? I can't answer all these things right now. Question in back. The short answer is yes. Some of these other gene mutations, the p53 gene mutation, which is associated with a rare syndrome called the Leigh syndrome, or the p10 syndrome, again give rise to other malignancies that typically affect younger, not older people. Almost all hereditary things arise in younger people, not older people. Yes?
You were saying something about the link between 6 to 8% chance if you have BRCA1 or 2 for breast cancer, something like 40% for ovarian cancer. Is it, would you necessarily get both?
You could get both. Because most women do pretty well with breast cancer, the more common story is to have breast and then ovarian cancer. Ovarian cancer tends to be a more ominous disease when found. Obviously, if you die from ovarian cancer, you're not at risk for breast cancer. But you can get both. And that's one of the things we're finding is more and more women get screened for these tumors. Yes.
But wouldn't she, if she had a gene, wouldn't it become for, let's say, breast cancer too?
Wouldn't it be?
She's just as susceptible for ovarian cancer as breast cancer, and she had her uterus hysterectomy.
Yes, so obviously that's right. So a hysterectomy and oophorectomy, removal of her ovaries, would not protect against breast cancer. Because we have some screening strategies for breast cancer, exam, mammogram, MRI, such women often elect to have the ovaries taken out but to retain the breast because that's both publicly visible and we have some better screening methods. Other women might elect to do prophylactic surgery removing all those organs. Again, I just want to come back to this point that the interesting thing about these tumors is they actually seem to be a little bit different at the molecular level than non-BRCA1 and 2 associated tumors. This is not a one-to-one concordance. We certainly see basal-like tumors that are not in BRCA1 and 2 populations, but clearly these basaloid tumors that are ER negative, HER2 negative are more common in particularly the BRCA1 patient population. Well, the first question is, how should we treat the breast itself, and should we leave the opposite breast intact in a woman who has BRCA1 mutation and who gets a breast cancer? And these are data from the group at University of Michigan, where they followed women who had one breast cancer and looked for cancer in the opposite breast. So if you wait 18 years, there is some risk, about a 10% risk of a second breast cancer in the opposite breast if you have an isolated sporadic breast cancer. By contrast, there's nearly a 40 or 50% risk over time of getting a second breast cancer in the opposite breast for women who have a BRCA1 or 2 mutation. So that means that we are frequently considering opposite-sided breast treatment in women who have one breast cancer if it's BRCA1 or 2 associated. Otherwise, it must be said that tumors tend to behave fairly the same in sporadic and hereditary cases, and the other principles of treatment in terms of chemotherapy and hormone therapy are more or less the same. So what can we learn, or what can patients learn from genetic testing? Well, consider this family where the husband does not have has a normal BRCA1/2 complement, and the wife has a mutation in the BRCA1 gene. Again, these are autosomal genes, so it could be the other way around. And in fact, a lot of patients come in and they say, oh, well, nobody in my mother's family had breast cancer, but in my father's family they all did, but I know I'm safe. It doesn't work that way at all. These are autosomal genes. Now, if you're twin sisters or a daughter of this couple, you know, you're thinking, I have a 50-50 chance of inheriting this gene from my mother in this case. Now, some women want to know this information and some do not. You could test these women, and it might turn out that the sister here has a normal BRCA1 and 2 inherited from her father, and therefore her lifetime risk of breast cancer is 11%, the national average. By contrast, this woman might have inherited a mutation and now faces an 87% risk of breast cancer. And it's easy to imagine that with those kinds of numbers, people could at least make better decisions for themselves about how best to proceed. Now, it must be said, it's very interesting. Not everybody wants genetic testing, and not everybody makes the same decisions with genetic testing. But these are the kinds of issues that genetic testing can help clarify. At least for an individual person, you find out where your risk shifts towards. Again, what are the implications? A positive test would tell this woman she's at increased risk of cancer and would also suggest that other family members might be fruitfully tested for themselves. A negative test depends on what your family background is. A lot of families have a relatively high family history, but if they're known to be BRCA1 and 2 negative, then a negative test in the next family member obviously is not informative because we don't know why they're having this genetic risk. By contrast, if you come from a family that is known to have a BRCA mutation, and you're negative, well, that often provides you with great reassurance because you know that, in fact, your risk is much more that of the average population and not somewhere between a 60 and 80% lifetime risk of breast cancer. And finally, if you're not known to have a BRCA mutation and it's a negative test, while it's possible it could be a false negative, it's very unlikely to be because the distribution, according to Bayes' theorem, you're familiar with Bayes' theorem, testing is a function of the prior probability. If it's already a low risk to begin with and you get a negative test, the overwhelming likelihood is that it's a true negative test and you can provide further reassurance to those family members. So the benefits are that it can provide relief from uncertainty, explain cancer or not within a family, and assist management decisions. This might also lead to prevention. And there are a variety of prevention strategies that have been studied for women with BRCA1 and 2 predispositions. Obviously not something we usually recommend as a preventative strategy, but having multiple children does seem to protect from breast cancer. And there's also evidence that the drug called tamoxifen might reduce the risk of breast cancer. Now, tamoxifen has been studied. It's an anti-estrogen medicine. It's been studied in women at high risk for developing breast cancer, and it reduced by about 50% the risk of both invasive breast cancer and DCIS in a very large study looking at women at high primary risk, 13,000 women. Now, the interesting thing about using an anti-estrogen therapy is it only prevents estrogen receptor positive breast cancer. So in that same study where women got either a placebo or tamoxifen, they had the same risk of estrogen receptor negative breast cancer. So an anti-estrogen doesn't affect your primary risk of ER negative breast cancer. But the missing tumors, the reduction in tumors with tamoxifen, were all to be found in the ER-positive subset of breast cancers, which entirely fits with the targeted and known effects of tamoxifen. Now, the other thing that patients can think about is prophylactic surgery. There actually have not been prospective randomized trials, because you can imagine you can't get women to be randomized into a prospective study of whether they should have bilateral mastectomy or not. But in a large retrospective series from the Mayo Clinic, they followed the experience of nearly 650 women who underwent bilateral surgery because of a strong family history. Most of these women were treated in the 80s and early 90s before we actually had genetic testing. They stratified these families into low risk and high risk. They predicted that in the low risk group, they would see 38 breast cancers among these 640 women using mathematical models that exist. And they only saw 4. You ask, how can you get any breast cancers in women who've had bilateral mastectomy? It's because sometimes when you do the surgery, you actually find the breast cancer occult in the breast when you do the surgery. Sometimes it's because, for technical reasons, the entire breast tissue isn't removed and there's some residual focus of breast tissue at the margins. And similarly, though, in women who are at much higher risk of breast cancer, again, nearly a 90% reduction in the expected risk of actual breast cancer cases. So it sounds pedantic, but mastectomy is highly effective preventative therapy for these tumors. The other thing to think about, as was alluded to by the question, is prophylactic oophorectomy. And again, in a non-randomized trial amongst BRCA1 and 2 carriers, about 100 women elected oophorectomy, and about 75 women elected observation for about a 4-year period. In this group of women who had oophorectomy, there were 3 breast cancers and 1 case of peritoneal cancer, which is a variant of breast cancer arising from the normal linings of the body. By contrast, there were 4 women who had ovarian cancers and 1 of those peritoneal cancers. So again, about an 80% reduction in risk by removing the ovaries in these women. And finally, in a woman who has 1 breast cancer, the question is it worth worrying about second events, or is she more likely to succumb to her primary tumor? And there have been some very nice mathematical models where they've tried to estimate the life expectancy gains from doing prophylactic surgery in women who've already had breast cancer. And not surprisingly, the prophylactic gain depends on how bad your first breast cancer is. If you have a good prognosis primary breast cancer and you're not likely to pass away from this breast cancer, then removing your bilateral mastectomy gives you an extra 2 years of life, Oophorectomy gives you an extra 2 years of life. Together, they give you an extra 4 years of life on average. By contrast, if you have a high-risk breast cancer, your risk of dying of that breast cancer that we already know about is higher, and therefore the relative benefits in terms of life expectancy gain are lower. Having said that, for many women with good prognosis breast cancer, we are thinking about these prophylactic surgeries. And so you can imagine that finding out the genetic information can be very important to women. And the current way we approach most such women is that if we know they have a BRCA1 or 2 mutation, and they've either not had breast cancer or they have a low-risk breast cancer, we usually recommend removal of the ovaries once they're done with their reproductive lives, because that's so hard to measure clinically, it's hard to detect at an early stage. And then we recommend either very close surveillance of the breast with ultrasound, MRI, and physical exam and mammogram, perhaps in addition to tamoxifen, which obviously leaves the breast intact, and we have pretty good ways of early detection there. Or if the woman prefers, or if it's difficult to do imaging in her, we recommend prophylactic mastectomy. So let me wrap up the moral of these two stories and then take a couple more questions before Dr. Brugge comes to the podium. Again, the points I wanted to emphasize from these two vignettes, which were that we are using the biology and a sophisticated understanding of acquired genetic change or hereditary genetic change, not just to understand intellectually, why did this happen, how did it happen to me, what does this mean, the sort of phenomenology that we all appreciate, but actually to treat cancer patients and to make real decisions with real individuals. The considerations I find are surprisingly powerful, but also surprisingly complex. And especially given the little level of scientific literacy, and I really would underscore amongst both physicians and amongst patients. Having said that, I am often impressed that patients understand the key concepts. I mean, they know what it means to have a gene that can give rise to breast cancer, or to have a tumor that is sort of HER2 positive versus HER2 negative. And I think the challenge that we have as providers, and the challenge that our patients want now that, you know, they read the Wall Street Journal of Medicine and every week there's some new discovery that's on the front page of the Times or in the Globe, is that we clearly need a more sophisticated understanding of science in the public, not because it's sort of interesting in the way that I find astronomy to be interesting, you know, and that lunar eclipse was great, you know, 2 weeks ago, but because this is actually going to affect how people and their relatives are treated. And this is going to be the case for lots and lots of tumors and hopefully other diseases as well. Breast cancer has really been a paradigm for this in the solid tumor field and has led the way. But the point is, it's not just some abstract thing. This is something that's really patients are going to need to understand so they can understand their disease and their treatments. So I'll conclude there. Thank you very much for listening.